Hello, good afternoon, everyone. I hope you had a good weekend. We will be finishing up lecture two today, hopefully. But before we get started, let's quickly review some of the key concepts we learned last week. So we talked about how all the different types of macromolecules, the carbohydrates, the proteins, the nucleic acids, with the exception of lipids, they are all made up of individual building blocks that repeats themselves. And we call these building blocks monomers. So carbohydrate is made up of carbon, hydrogen, and oxygen. And the monomers that build the carbohydrates, they are called monosaccharide. And we learned three types of monosaccharides for carbohydrates. We have glucose, we have fructose, and we have galactose. Okay. So these individual building blocks can be connected together through a type of chemical reactions, and you can create a disaccharide. A disaccharide would be double sugar, two of these things connected together. So if you take a glucose and you link it together with another glucose, then you will get a maltose. And in the process, you will remove a water molecule. So these kind of reactions, they are called dehydration synthesis, right? So, if you connect them together, it's called dehydration synthesis. Okay. Similarly, you can take glucose and you can connect it with a fructose and you will get sucrose. We did all these already, okay? Like I'm just reviewing for you to kind of get you all warmed up about it. So this is sucrose. And the last one here, glucose plus galactose. Does anybody remember what that gives you? Glucose plus galactose. What are we going to get? That's right, Melissa. That is lactose, which is found in dairy products, right? Okay, so all of these are examples of disaccharides. Okay, and then we also talked about polysaccharide. Polysaccharide. And polysaccharides are basically long chains of monomers connected together to form the carbohydrates. And the example I gave you are all polymers of glucose. It's just a bunch of glucose connected together, but they are different in how they are connected together. So we have things like cellulose. Cellulose would be structural support in plants. It forms the cell wall and the fibers. We have another one, which is chitin. This is a modified glucose monomer with a bit of nitrogen, and this forms exoskeleton of insects and crustacean like crabs, lobsters, that kind of stuff. There are two more. Anybody remember? What are some additional polysaccharide that we learned about? Okay, good. Starch is one, and that is the storage form of energy in plants, okay, in roots and stem. And what about us? What about humans, animals in general? Where do we store our extra glucose? That's right, Naja. It is going to be glycogen. Glycogen. Storage form. And what organs do we store them in? Liver, yeah, and muscle as well. Okay, liver and muscle. Okay, so that's what we learned last class, amongst other things. Hopefully that's a good summary for you. Um, and please start studying. Your first quiz will be next Monday. Okay, next Monday. We still have class, okay? You got to come to class as well, but then you also have to do the quiz. I'll tell you more later. All right. Okay, so today we will start with lipids. Instead of doing the PowerPoints first, let me give you the summary first, and then you can have the summary next to you when we are going through the slides. Hopefully, it'll make more sense that way. So lipids. First of all, lipids are a little bit different compared to the other three kinds of macromolecules. Okay? Carbohydrates, proteins, and nucleic acids, they are all made up of individual monomers. Okay, and the monomers form very long chain, like a train. Okay, but lipids are not very long chain. It's not a polymer. Right? Instead, it's made up of components that you put together. Right? It'll make sense after I give you the summary. Right? So put lipids in the middle, and then I'm going to show you the different kinds of lipids. Okay. So the first kind of lipid is fat. Fat has a fancy name. It's called triglyceride. Yeah, very good. Triglyceride or fat. And please use some colors. Fat is made up of this thing called glycerol. Please do not confuse glycerol with glycogen. It's a common misconception for people. Okay, they have nothing to do with each other. They just sound very similar. Okay, so there's this glycerol. You need one of these glycerol, and you are gonna have these fatty acids. Okay, fatty acids. So it's not a bunch of glycerol connected together and form a polymer. It doesn't work like that. You just need these components. To build a fat, you need one glycerol and three fatty acids. That's the recipe, if you would, to make fat. Okay, so fat is for long-term storage, long-term energy storage. It is for insulation and protection of organs. Okay. Insulate means it keeps them warm. Like today, it's like what, feels like minus 27, the forecast says. And you want to keep your internal organs warm, right? A little bit of fats is going to help you do that quite well. It's going to protect your organs. If you fall, the fat acts as a little bit of a bubble wrap. It cushions the organ, so it's not going to damage them. Right, so that's triglyceride, okay? Now we're going to do another one here. It is called phospholipid. Phospholipid. So phospholipid has similar components. You're gonna have the glycerol. And again, I know I say this all the time, but please try to use color and follow along. The notes will make so much more sense if you do that. Okay? And then we will have these, zigzag lines. And what do we call them? Same thing over here, right? Fatty acids. Okay, but this time you only need two. Okay, so three fatty acids here, two fatty acids. And then you need one more thing here. You need one more thing. It's called the phosphate head. Okay, phosphate head, phosphate group, doesn't really matter. So what are phospholipids? They are component, major ingredient of the cell membrane. Okay, we haven't really learned cells yet, but cell is like, it makes up the whole living organism. We briefly talked about it, I guess. And if you draw a cell, right, the the outside, the surrounding of the cell, that's called a cell membrane. Okay, and then you have like a bunch of other stuff in it. So the cell membrane is made up of this thing called a phospholipid. This is just a summary, okay? Like, don't worry if you don't 100% get it yet. Just putting everything here for you so that you have a sheet next to you and you can refer to it when we are going through the PowerPoints. All right? We have another lipids here, another kind of lipids, which is called steroids. Steroids. Right? So steroids are, I mean, like people hear the word steroids, they might think performance enhancing drug, but not necessarily. Your body has different kinds of steroids. And to make a steroid, you need carbon rings. Okay, so these are carbon rings, and there are always going to be four of them. Oops, it doesn't really matter. You don't have to, draw them exactly as it is. So four carbon rings, and they are fused together. They're stuck together. And on the carbon rings, you have different accessories. Okay, I'm not gonna make it too complicated for you, but these little purple things that are sticking out, we call them functional groups. Functional groups. They are like add-ons to the core structure. And depending on what kind of functional groups you add to the molecule, it will give it different properties. Okay? Functional groups, you can think of them as add-ons. Okay? Like when you buy your phone, like do you want a, screen protector? Do you want a case? Do you want one of those lanyard things that you put on, or like a ring at the back, or all these extra things that you can add to it? It's going to add functionality to it, right? And that's what these functional groups are. And what do steroids do in the body? Also part of the cell membrane, and they form hormones. Hormones, right? E.g., sex hormones. Does anybody know the name of any sex hormones, either in guys or girls? What are Okay, I have an answer already. Very good. Estrogen, yeah. Testosterone, good. Progesterone, right? And all these are steroid hormones. Okay, that's steroid. And then we just have one more kind. One more kind right here. Yep, progesterone, very good. This is wax. Wax. Okay, we don't really have wax. I guess we kind of do like our hair has a waxy coating to our skin. It has kind of like a waxy coating to it to prevent water from just like going right through. And wax is basically here it is again, these, you know, uh, long chains of zigzag, which we call fatty acids. Fatty acid. And there are like functional groups attached to it, but this is like a special kind of functional group, okay? It's OH, and you will learn this in chemistry like next semester. If it has an OH attached to it, we call it an alcohol. Okay, not necessarily like drinking alcohol. Like drinking alcohol is a type of alcohol that has OH, but there are like different kinds of alcohol, right? Like your windshield fluid, that's actually a kind of alcohol, right? It's you cannot drink that; it's gonna poison you. But chemically, it has OH in it, and that's what makes it an alcohol. All right, so wax is kind of like that. It's these fatty acids with some alcohol stuck to them, and the main function of wax is to form water resistant barriers. Think about the surface of an apple. It's like waxy, shiny, right? And if you put water in, put apples in a bowl of water, water is not going to go in the apple because of the skin. And there you have it. The four types of lipids. Um, just keep it next to you. Like I said, I'm going to go through the PowerPoint slide now, and you can refer back to it, to these things as we go through it. But any questions so far for lipids? Okay, very good. And I'm very happy to see all of you still coming to class. Okay, it's week 3 and attendance is still looking good. So that's very good. Okay, very good habit, very important to be in class. Although I want to remind you to work on the self quizzes. Okay, I see that not a lot of you have had a chance to do that yet. Please try them out before the actual quiz next Monday. It will help you get used to taking tests in an online environment, and more importantly, you will have a chance to install the lockdown browser, which is mandatory for all the assessments you'll be doing in the course and some other courses as well. Okay, chemistry and math, for example. All right, let's move on to lipids. Uh, just like carbohydrates, it contains carbon, hydrogen, and oxygen. Okay? And there are trace of phosphorus in some type. Okay? Trace of phosphorus. Unlike other macromolecules, lipids are built with components, which is what the summary tries to show you. It's not a long chain. It's just, parts coming together. And all lipids are hydrophobic, meaning they do not dissolve in water. Okay, it's opposite of hydrophilic. Carbohydrates are hydrophilic. Right? If you think about a spoon of sugar, it's going to dissolve in water, no problem. Not lipids, though. Okay, they are hydrophobic. Let's take a look at fat. Fats are also called triglyceride. We wrote this down already. It's made up of one glycerol and 3 fatty acids. Okay, the glycerol looks like this. It has 3 OH on it. And if you're thinking, hey, does that make it an alcohol? Yes, it does. Okay, anything with OH on it like this is an alcohol. And over here, we have 3 fatty acids. Okay, now we're going to talk more about fatty acids later. It turns out some fatty acids are saturated, others are unsaturated. And you can tell by looking at uh, the kind of chemical bonds it contains. But one glycerol plus 3 fatty acids, if you do dehydration synthesis, You'll remove a water molecule here and another one here and then a third one here, right? Then you will be able to connect them together and form a fat molecule. Okay, so you need to make three connections. That's why there are three water molecules um, released. Yeah, so they're good for long-term energy storage. If you are fasting for more than a few days, then the reserve in the liver is not going to be enough. Then you got to tap into your fat reserve. Okay, you're going to start burning up some of the fats as fuel. And like I said, they're good for insulation and protection of organs. Where do we get our fat? Usually people don't really have to worry about, deliberately consuming fat, especially, in Western diet. It's like there are lots of fat everywhere. But butter would be a source of fat, oil, of course. And there are some good fats as well, right? Fish has good fats in it. Things like walnuts and even certain kinds of vegetables, they have high good fat content in it, right? Like avocado, if you think about it, that's a fruit that has a lot of oil content in it. Now, when it comes to fat, it depends on what kind of fatty acid it contains. Remember, it has three fatty acids, right? Three of those zigzag tails. Now, those tails can either be saturated or unsaturated. If all the tails, all three of them, are saturated fatty acids, then the whole fat molecule is called saturated fat. Right? On the other hand, if even one of the three fatty acids is unsaturated, then the whole fat is going to be unsaturated. All right, so let me repeat that one more time. There are three fatty acids. If all of them are saturated fatty acids, then the whole molecule is saturated. On the other hand, if even one of them is unsaturated fatty acids, then the whole thing is unsaturated fat. It's possible for you to have two unsaturated fatty acids or even three unsaturated fatty acids, but doesn't matter. If you just have one, then it's already whole thing is being called unsaturated fat. Now, which one is healthier? What do you guys think? Saturated fat or unsaturated fat? Let me do a poll here and see. Okay. If you need to consume some fat, which one is relatively healthier? A, saturated or B, unsaturated? Which one is better for your body? A for saturated, B for unsaturated. Very nice. Anyone else? All right, we are 50-50 dead in the center. Half of you think is saturated, half of you think is unsaturated. Let's take a look at this. So, what makes it a saturated fat? It turns out a saturated fatty acid is a fat that has a maximum number of hydrogen connected to it. Okay, so if you look at the fatty acid tail, the zigzag here, it's made up of two things. Right? It's made up of carbon and hydrogen. And you will learn this in chemistry probably in the coming weeks. But if you have a carbon atom, it can form at most four connections with other atoms. Okay, so like if you have a long tail of C's, it's going to form a connection with another C here and another connection with another C here. And then it will connect with hydrogen and hydrogen. This carbon will also connect with hydrogen and hydrogen, and then so on and so forth, right? It just keeps on going and going. And in saturated fat, right, it has no more rooms for any more hydrogen. Because if you look at the carbon atoms, it's full. It has four connections, and four is the maximum number of connections it can make. So even if I want to put more hydrogens onto the carbon, it literally won't fit. I cannot put like a fifth one there, right? It violates the rules of how chemistry works. So only four, and there's no more room for hydrogen. And that's what saturated means. It's saturated with hydrogen, right? It's connected to the maximum number of hydrogen. And that makes the tail just go up and down, zigzag like that. Now, if you look at a fat molecule, there is the triglyceride. I know it goes zigzag, but I'm just going to draw them straight down like that. If you put one fat molecule next to another fat molecule, because the tails are all relatively straight, it's very easy for you to pack them close to each other like that. I can put another one right here, and the fat molecule just clumps together easily. For that reason, they are solid, mostly solid at room temperature. Okay? And if it's easy to pack together at room temperature, it's also relatively easy for them to pack together in your arteries, for example. Right? And so if you consume a lot of saturated fat, they will slowly build up inside your blood vessels and slowly cause them to clot, right? And that is not a good thing, of course. Most animal-derived fats are saturated, right? So we're talking about butter, we're talking about lard. People use lard to make pastry, right? It's very tasty, it's very fluffy in the end, but it's very, unhealthy in a way, right? So these things are saturated. Unsaturated fat is made up of glycerol and at least one unsaturated fatty acid. So in this case, and again, it will make more sense later when you learn this in chemistry, but in this case, the carbon is going to be connected to another carbon here. I'm not going to ask you to draw these things. The key thing to know is that instead of connecting just once to another carbon, it will connect twice. Okay? Sometimes it will connect twice. And so instead of making four connections, it's a one, two, three connections here, which means there are still room for more hydrogen. It's possible we add another hydrogen here, and then we break this bond here, right? And then we add another hydrogen, right? So because there is still room for hydrogen, we say it is unsaturated fat. And this thing here where they share twice, there are two connections between them, we call them a double bond. Okay. So anytime you have a double bond in a structure like this, it causes the fatty acid tail to create a sharp turn. So if I draw the fat here, the unsaturated fat, you will have two straight one in this case, and then the third one would like stick out like that. Okay? So because it's sticking out, it's very hard for another molecule to get close to it. Okay? Imagine lining up uh, for something. Okay, lining up at the grocery stores, but everybody's like sticking their arm out and sticking their feet out. That prevents people from packing too closely, right? And that's exactly what happens here. If they don't pack as closely together, then they are liquid at room temperature. Most plant-derived fats are unsaturated. Okay, so give me some examples of fats in your kitchen that's unsaturated. They are derived from plants. Anybody have examples for me? Yep, olive oil, that's a good one. Anything else? What kind of oil do people cook with other than olive? Yep, avocado oil, right? Grapeseed oil, canola oil. There you go. Okay, you name all of them. Very good. Now, remember, there are exceptions, right? So we try to put them in categories as much as we can, but sometimes there are exceptions. And I was waiting for someone to say, and there it is in the chat, right? Someone said coconut oil, right? So coconut oil, even though it is a plant-based oil, if you look at it, it's actually semi-solid. Okay, so coconut oil has a high saturated fatty acid content in them, and that's what makes them a little bit solid rather than liquid. So not always plant fats are not always unsaturated. There are exceptions to them. Are we clear on the difference between saturated and unsaturated? Right? It all has to do with the kind of tails they have. You are basically looking for the double bond. The double bond creates a kink, and that makes them harder to pack together. Okay, if you got to chuck down some oil, unsaturated fat is the better choice. Even so, you're not supposed to have too many of them, right? Okay, there are some absolutely important fats that we need to have. We call them essential fatty acids. These are fatty acids that are necessary for normal bodily functions, and we cannot make it in the body. The only way to obtain them is to eat them. Right? We call them essential fatty acids. Examples of these include omega-3, and there are different kinds of omega-3. The common ones are called EPA and DHA. If you are pregnant or if you have kids, then you would know these are often prescribed as supplements during pregnancy because the EPA and DHA are important for brain development in the babies. Okay, so like if the mother is taking these supplements, it goes to the babies and give them healthier brain development, I suppose. That's what I'm trying to say. Other than omega-3, you probably heard of like omega-6 and omega-9, right? Those are also essential fatty acids. Okay. Your body needs them, but you can't make it. So you need to eat them. Right. So fish has good fatty acids, the omega-3, 6, 9, avocado, nuts, all these things are very rich in these essential fatty acids. That's what they look like, the EPA and the DHA. The reason they're called omega-3 is because the double bond occurs on carbon number three. Okay, the first double bond occurs at carbon number three. Right? You count one, two, three, right? If it's omega-6, then the first double bond is going to happen at carbon number six. Right? You don't have to know this particular slide. I'm just showing you in case you're wondering why they're called the way they are. Okay, and you don't have to memorize these names as well. EPA, DHA, it doesn't matter. Okay, just know that, just understand the meaning of essential fatty acids. That would be enough. Okay. Besides fetal brain development, the omega-3, 6, 9 are also good for heart health, right? And some adults, even if they're not pregnant, they might be taking these supplements. Okay, so just to give you another visual side-by-side comparison, both saturated and unsaturated fats, they have the glycerol. That's what this part is. Glycerol. And then these things are fatty acids. And you can see in the saturated ones, there are no more room for hydrogens, no double bond, no kink, no bend. It's all straight. This guy, the unsaturated one, all three fatty acids are unsaturated. You can see double bonds here, double bond, double bond. So you can see the bending and the kinks in all three tails. And again, it doesn't have to be all three. Right? If only one of them is unsaturated fatty acids, then the whole molecule is unsaturated. Okay? If it's all three, then we actually call it polyunsaturated, and that tends to be like the healthiest kind of fat. Okay, there is something called trans fat. Trans fat is a byproduct of a process called hydrogenation. Okay, so unsaturated fat is healthier, but then it doesn't make sense to put unsaturated fat on your toast, for example, right? Like you cannot just drizzle olive oil on your toast and then eat it. It would be like, soggy, right? Although, in some cuisine, Italian, for example, like I think they dip it in the oil, but like everyday morning, you're not gonna do that. So, um, what we do is we spread things like margarine on them, right? And so what is margarine? Margarine is basically unsaturated fat, and you forcibly add hydrogen to them. Okay, that process is called hydrogenation. So unsaturated fat, and then you add hydrogen to it under pressure, high pressure, and high temperature, and then it becomes saturated. Okay, or at least part of it becomes saturated. And then you can spread them. Okay, so the concept is if we start with a healthier fat, then it's better than just spreading butter directly onto your toast, right? But the problem with that is this process generates a byproduct, which is trans fat. And trans fat cannot be metabolized by the body. You don't have the enzyme to break it down. You don't have the means to break it down. So it just kind of accumulates in the body. And it's really bad, trans fat. It increases the risk of heart diseases and diabetes and obesity significantly if it's being regularly consumed. And so Canada actually banned artificial trans fat in 2018. Right? So they used to have a lot of trans fat in fast food, right? Because it makes them taste better and it also increases the shelf life of some products. So not only in fast food, but in processed food, there are a lot of trans fat in it. But Canada, the government has banned it since 2018. So it makes them slightly more healthy. You can read more about it in this news article link right here. All right, that's it for fat for triglyceride. Okay, if you still have the summary with you, that's one down, three to go. Next, we're gonna look at phospholipid. Phospholipids. Okay, so phospholipids are they are the key component of cell membrane and it's made up of three things. There is the glycerol here. It's hard to recognize it when it's all like written in CH and O, but there it is. That's the glycerol. It's the same glycerol as the fat. Okay. There is the phosphate head or phosphate group, and then it has two tails, two fatty acid tails. Okay. We'll talk more about this later in another lecture, but the tails can be like this one here. Do you think this one is saturated or unsaturated? The one on the left. What do you think? Just look at it compared to the one on the right. What do you think this one is? Do you think it's saturated or unsaturated? How do you tell? Yeah, okay, so a couple of people say this is saturated, right? What gave it away? How do you know this is saturated? Yeah, it's all aligned. It's all straight, right? There's no bend to it, no kink, no double bond, right? And if you look at this one, there's the double bond, there is the kink, the bend, and that would be an unsaturated, right? Unsaturated fatty acid. So it doesn't really matter for the phospholipid. They can both be saturated. They can, be one of each, and they can both be unsaturated. And we're gonna talk about the implications of that at a later lecture. Okay, but the key things is that it has a polar head, it has two tails, and it has glycerol in the middle connecting them. So in addition to CH and O, there is a little bit of phosphate as well, and the phosphate is right here in the head. Okay, now that makes this molecule very unique in the sense that the head is, oops, the head, because it's made up of the phosphate and some oxygen, the head is hydrophilic. But the tails are, okay, hydrophilic head. The tails are hydrophobic tails. So the head binds well to water, but the tails will avoid water. Okay, so these kind of molecules are very good for forming the cell membrane. I'll show you that on the next slide. But if you use detergent, that's actually very similar to this phospholipid molecule. Right? If you try to wash a greasy dish with just water, like doesn't matter how long you wash it, it's the grease kind of still stick on, right? You're not going to be able to get rid of it easily, and that's because water hydrophilic and the grease is hydrophobic. They don't mix together, right? But when you add the detergent is like this molecule. It has a hydrophilic head, which binds to the water, and it has the other side, which is hydrophobic tails, and it will bind to the grease, and it kind of, merge them together, blend them together, and it helps you get rid of the grease from your plate. Okay. So these kind of molecules are quite useful and unique because of their dual nature. And one part is water loving, the other is water repelling. And so the cell membrane is made up of two of these phospholipid layers. Okay. It looks like a sandwich, and all the tails are in the middle, and then the hydrophilic heads are either facing outside or inside. Don't worry about it right now. More on this later in lecture 4. Okay, we're going to learn more about the cell membrane in greater details uh, in lecture 4, like I said. All right, I'm going to pause for a moment. Anybody have any questions for me? Do you understand everything I've said? Okay, I will move on then. The third type of lipids would be wax, and wax covers exposed surface of plants. They help protect and lubricate skin and hair. If you drop some, if you put your hands together and put some water in it, water is not going to go through your skin and into your hand because of the wax. Okay. And you will find wax in other structures like honeycomb. People use bee wax to make cosmetics, right? Uh, and the components to make up the wax includes the fatty acids, and there are some alcohols connect to it. Okay, that's all you have to know for wax. Okay, nothing too crazy. Finally, we have the steroids. Okay, steroids have this ring structure to it. There are four rings always in steroids, and there are different things attached to it. Okay, like here you see another alcohol attached to it. There are some other extra things that are attached to them, like here and there to the core ring structure, right? And depending on what additional functional groups you have, it would give it different properties. For example, the alcohol here is gonna make it a little bit more hydrophilic, right? And then you have this long tail here that would allow it to maybe perform certain signaling functions, right? It really depends on the kind of functional groups you are talking about. So this particular molecule actually is cholesterol. And cholesterol is not necessarily bad. Sure, it's something you have to monitor when you do your blood test, but there are good cholesterol and bad cholesterol. And actually, you need to have cholesterol in your body for making different hormones. Okay. So not only will you find cholesterol in the cell membrane, and you will also use cholesterol to make various steroid hormones. Okay, so we already talked about estrogen and testosterone. Now you can see the rings are there. You always have the rings, but the stuff that are attached to it is going to be different depending on what kind of steroid you are talking about. I do want to mention something here. Not all hormones are steroid necessarily. Okay, there are different kinds of hormones. If you don't know what hormones are, they are signaling molecules in the body. Like if your brain wants to communicate with the thyroid or something, then it's gonna send some hormones to do it. Okay, if your if your brains want to communicate with the ovaries, again, it sends hormones as chemical messengers to communicate with them. Some of these hormones are steroids. Others, as you will see, are proteins. Okay, so there are different kinds of hormones in your body. And that is it for lipids. Let's quickly do a few poll questions here. Hopefully, we'll get more participation, and we'll move on to another um, topic after. Okay, that's good. Okay, there's a question in the chat. Are steroids always hormones? Not necessarily, okay? Some steroids are found in the cell membrane, and those are not hormones. Okay, so steroids are not always hormones, and hormones are not always steroids either. Okay. All right, I'm gonna close the poll. Anyone else? Last minute attempt? Okay, this says glycerol with three fatty acids. All you have to do is look at the summary note here. You can see the glycerol and the three fatty acids, and the answer would be triglyceride. Okay, triglyceride. Next one. We're looking for the false statement. Okay, give this a try. I'll be right back. Okay, not a lot of responses coming in. Please do participate whenever you can. This is a great way for me to see whether you guys understand or not. The polls are anonymous, so you don't have to worry about that. Which of the following is false about lipids? So let's see. Steroid has at least one fatty acid tails. That's not necessarily true. Okay, if you look at the notes we wrote, steroid has the four rings, and that's about it. Okay, the other stuff that you attach to it might not even be fatty acids. It could be just alcohols or like other functional groups that you haven't learned yet, but it doesn't necessarily have to have fatty acid tails. Canola oil contains unsaturated fatty acid. That's true. Okay, plant-based. It's mostly unsaturated. Triglycerides store energy. They do long-term storage. Cholesterol is a steroid. It literally says so in the notes. And glycerol is a component of triglyceride and phospholipids. That is also true. Okay, take a look here. Both triglyceride and phospholipids, they have the glycerol, right? Which is why it's important to color code your notes. It just makes it much easier to see them. So the correct answer for this question is A. All right, we're going to move on to proteins next. Anything else you want to ask before we do that? Proteins is the hardest one, right? Uh, no, no questions? All right. Chemistry of proteins. Right? So what are the main elements that makes a protein? Again, you have the CHO trio. These three things are like always there. Okay? It's there for carbohydrates, there for lipids, and it's here for protein once again. Mainly composed of CHO and nitrogen as well. Nitrogen. Okay? And then there are traces of S. Trace means a little bit, right? A little bit of sulfur. The monomer of proteins, so now we are back to the individual building blocks, okay? They are called amino acids. Amino acids. There are 20 amino acids that are found in proteins. 9 of them are essential. What does essential mean? Does anyone remember? What do we mean by essential amino acids? We talked about essential fatty acids earlier, right? So essential amino acids, it's the same meaning, right? Your body needs it, but you cannot make them. Okay, so these amino acids, you have to consume it from your diet. Okay, and here they are. You don't have to memorize them. Do not memorize them. Okay, we have histidine, valine, tryptophan, so on and so forth. These are essential amino acids. And if you are not a particularly picky person, if you kind of eat, a variety of food, then you would be able to obtain them, just through your, you know, current diet. Okay. There are certain foods that are particularly rich in certain kind of amino acid, like phenylalanine is particularly rich in avocado, beans, rice, so on and so forth. So if you are looking for, a boost in those kind of amino acid, maybe those are the food that you need to turn to. There are some conditions where a person is not able to break down certain kind of amino acids, and so you would try to avoid some of these food, right? One such condition is called PKU, which stands for phenylketonuria. A phenylketonuria. So PKU is a rare genetic condition that prevents the person from breaking down phenylalanine. Okay, you can see over here, phenylalanine is one of the essential amino acids. So if your body cannot break it down, then the phenylalanine starts to build up inside the body. And if this happens in a baby, then it's going to interfere with brain development. So one of the first things that they screen for in newborn is PKU, amongst other things. Okay. They basically collect a little bit of blood sample from the baby. They do what's called a heel prick. Just cut the heel here a little bit, just a drop of blood, and then they go test it, and they will know whether the baby has PKU or not. If the baby has PKU, then they cannot drink the breast milk or regular formula, okay? Because those contains phenylalanine, and like I said, it's gonna be a problem for brain development. So they have to put on a special type of formula, right? And to make sure that they are not ingesting phenylalanine. Okay, it's a genetic condition, so like you're either born with it or not. It's not something you will get later on after birth. All right, let's draw the general structure of an amino acid. Here we go. General structure of an amino acid. All right. So it has a carbon in the middle, carbon in the middle, and it is connected to a hydrogen on one end. Right? And then on one side, remember carbon can make four connections, right? So one connection at the top. And then on one side, it is going to connect to this functional group that looks like this. If you haven't taken chemistry before, this might be a little bit confusing, but you will be learning how to draw these diagrams in the coming weeks in your chem class. So over here, this configuration, this carbon with the double bond with the O and the OH, this is what's called a carboxyl group. Carboxyl group, and it makes it an acid on one side. Okay. I'm not going to ask you to draw these things, but if I give you a picture and I ask you to show me where the carboxyl group is, you would be expected to, click on this portion. On the other side, it is going to be connected to another group here. So this group of atoms with the N and two H's, that's called an amino group. Amino group. So that's why it is an amino acid. Amino acid. 20 amino acids, all of them would have this basic structure. Right? It would have an amino group to one side, a hydrogen at the top, and then the carboxyl group here. Here, I'll draw a little bit better um, so that it looks more organized, I suppose. You don't have to change yours, okay? Like, it's fine. And then the fourth connection would be something called R. Okay? R is called a side chain, and it is unique for each of the 20 amino acid. Okay, that's what distinguish one amino acid from another. Right? That's what gives them different properties. Okay, so catch up, draw this down, and I'll show you what the amino acids look like. All right, so here are the 20 amino acids. I don't know if there are actually 20 here, but at least there are some of them. And you can see there is the core structure. Right? There's the C in the middle, there's the hydrogen, and then there's the COO. I know they put a negative here and then we put an H. Don't worry about it. It's the same thing for our purpose. I didn't want to put the positive and negative to confuse you. It's already very confusing, I know. But this here, that would be the R group. So for glycine, one of the 20 amino acids, the R group is just a hydrogen. For something like alanine, the R group is CH3. Okay, and you can see they can get more complicated. Okay, some of them is like CH2 with a ring, and then some of them is really, really long, and then they have positive charge to it. And the point, the key takeaway from this is depending on what kind of R group it has, depending on what kind of side chain it has, it would give the amino acid different property. Okay. Some of them are non-polar, some of them are polar. Remember, polar means slightly positive and slightly negative in different portions. Some of them are positively charged, some of them are negatively charged. Okay. And these different properties will ultimately change how the protein behave. And the implication is quite big. Okay. And we're gonna come back to this again and again later. All right. So quickly recap. Proteins are made up of amino acids. There are 20 of them. Right? 9 of them are essential. You need to eat them. And what are the key elements that makes up a protein? Right? You can just recall it in your head. You don't have to tell me. Right? What are the key elements that makes up a protein? Right? So we have the triplet. It's always CHO. Nitrogen, right? You can see the nitrogen here. And trace of, does anybody remember? Trace of what? A little bit of what? That's right. Trace of sulfur, not phosphorus. Okay. Phosphorus is for lipids. Okay. Trace of S, and you can see if you pay attention to each of the side group, this is the guy that has the sulfur here. Okay. There's the sulfur right there. There you go. Another one that has the sulfur. Right. So just a little bit of sulfur here and there, not a lot of it. Okay, let's take a break and then we'll come back and do more. We'll resume at 1:35. Okay, see you in a bit. All right, let's keep going. So let's take a look at how we connect two amino acids together. All right. Now this might be a little bit difficult for you to draw along, and I'm never gonna ask you to draw this, okay? But you should have an understanding of how it looks like for some of the questions, okay? So maybe you don't want to draw along, just watch and then do it later when you're watching the recordings. So here we go. We're gonna have one amino acid right here. And then it's going to have a side chain. And depending on which of the 20 amino acid we're talking about, the side chain would look different. So I'm just going to put R1 here for side chain number one. And thanks to the magic of copy and paste, we're going to have another one right here. But this time, we're going to have R number two. Okay, R number two, side chain number two. A different amino acid. It doesn't have to be different. It could be the same. But let's just make it different. And we're going to try to connect these two things together. So how do we do that? Well, to connect them together, it's the same process for all of the other macromolecules. You need to remove a water molecule. Okay, so that's called dehydration synthesis. You're going to take away an OH here, you're going to take away an H here, and a water will come out. Okay, and then what do you get in the end? And what you get in the end is like this. I'm just going to copy the original here, R1, C double bond O, and then you have a new bond. That basically connects the C here with the N, like this. This is R2, like that. Okay, plus H2O. And this bond that we just formed that connects the two amino acids together, we call it a peptide bond. Okay, a peptide bond. Now you can repeat this a couple of times. You don't have to draw it now, okay? Just take a look here and see if you understand, and then I will explain, okay? Again, you don't copy this down. Okay, like this. There we go. I know it's very long, but just watch. Okay, so this is R1, first amino acid, second one. This would be like a third one, fourth one, fifth one, and sixth one. Okay, we're gonna take away all these arrows. Right? Every time you see the C and the N, that's a peptide bond, peptide bond. And so we have 6 amino acids connected together. There should be 5 peptide bonds, right? 1, 2, 3, 4, 5 peptide bonds. Okay, and you will remove 5 um, water molecules. And what I want you to pay attention to is the fact that this is NH and then CHCO. And then you can see it just repeats itself. NHCHCO. 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 And so on and so forth. It just keeps on repeating itself, the same thing over and over again. And so this is what we call the peptide backbone. It's the same thing being repeated. All right. Doesn't matter how long your amino acid chain is. Right. That if it's 6 amino acids, 100 amino acids, the peptide backbone will be the same over and over again. And what makes it different is the side chain, right? So here you have side chain number 1, side chain number 2, side chain number 1, number 2, so on and so forth, right? And that forms like the whole thing here, the whole thing. This whole thing is called a polypeptide. Okay. So I don't know if you see the parallel here. I'm just gonna quickly write this down for you, and you can take a look at it later. Okay? So for carbohydrates, the monomers, the monomers, they are monosaccharide. Okay, and you have glucose, fructose, and galactose. For proteins, the monomers are amino acids, and there are 20 of them. You don't have to know the names. Okay? What about the polymer? The polymer. For carbohydrate, what do we call the polymers in carbohydrate? Help me out, please. Oh my goodness, what do we call the polymers in carbohydrates? Anyone? Yes, that's correct. It is polysaccharide. And now for proteins, what do we call the polymer? Polypeptide. Okay. So it's like similar, right? We cannot do it for lipids because lipids don't have monomers and polymers, right? They just have different components being put together. Are we together? Please let me know if you have any questions at all. Okay, let's go back to our PowerPoint here. When we talk about protein structure, there are 4 levels that we need to consider. Okay? And each level contributes to the overall shape and structure of the protein. So let's start with primary. Okay, primary, the lowest level of protein structure. The primary structure of a protein is the unique amino acid sequence in the polypeptide. Think about the amino acids as beads. Okay, so you have 20 different colors of beads, and you're trying to make a necklace using these beads. All right. So, you can put the red one next to the purple one, and then green is next, another green, and then yellow, so on and so forth, and then you're gonna get a necklace, right? So if you read the colors of the beads from one end to the other end, that sequence is the primary sequence. Right? Of course, when we are talking about the proteins, we are not actually saying the colors. We are saying the identity of the amino acids. So for example, if you connect alanine together next to a proline, and then the next one is going to be a leucine, and then maybe another leucine, right? So like you write them out, alanine connected to proline, connected to leucine, and then another leucine, and then all the way. If you read out each amino acid, the order that you connect them in, that is called the primary sequence of your amino acid, the primary structure. Okay. Now, these are not random. Which amino acids you put together is going to be determined by your DNA. Right? So your genetic code actually contains the instruction for building all the proteins in your body. All right, and we're going to learn more about that later on in lecture 7, I think. So that's primary structure. Next, we're going to have the secondary structure. All right, just give me a second. I usually have to have a little bit of a prop here to help people understand it. But I don't know if I have it today. Oh, there it is. There we go. Okay, so here, this little pipe cleaner thing, right? Like imagine you have the beads on it, okay? I'm not gonna put it on because it makes it cumbersome. Right, so you connect all the beads together, and remember I was telling you if you connect all the amino acids together, the backbone, the peptide backbone is the same, right? The only thing that's different would be the side chain, would be like the color of the beads, so to speak. Right? So what's going to happen is some parts of the peptide backbone, it's actually going to uh, curl together and form a little spiral. You see this spiral here? Right? So these spirals, they are called alpha helix. This is a Greek alphabet. It stands for alpha, alpha helix. So you can see in this ribbon model, you have the alpha helix there, and then you have another alpha helix here. The whole thing can have, multiple helices. And like I might form another one right here, right? So this guy would, have helix here and helix here. It's also possible for some parts to form helix and other parts is going to form these zigzags. So zigzag goes like this. It's just up and down like this, right? Zigzag as opposed to spirals. So these zigzag, we call them beta pleated sheets. Beta pleated sheets. Okay? So again, it could be only alpha helix, it could be only beta sheets, or it could be a mix of both. And what helps stabilize these structures together are hydrogen bonds. There are hydrogen bonds that are formed between the peptide backbone. Now, I told you before, we're going to mention hydrogen bonds again and again. And it's okay if you don't fully understand what it is. The key thing to remember is anytime I say hydrogen bond, it's basically some kind of attraction, okay? Like magnets that helps stick things together, right? So like if you imagine the helix here, to prevent the helix from collapsing on its own, right? And from help them stick together, you have some hydrogen bonds here, so on and so forth. Okay, same thing with the beta pleated sheet. All right, so that's the secondary structure. Now, if I draw it out on a piece of paper, this is pretty much 2D so far. You have the helix here, and then you might have some zigzag, right? And you would have the amino acids like that along the whole way. So that's primary structure and secondary structure. Now, why do we care, right? Like, who cares if it's alpha helix or beta pleated sheets? Like, what's the significance of that? Well, I'm gonna give you an example. Right? Uh, have anyone heard of mad cow disease? Mad cow diseases. How do you catch mad cow disease? Anybody know? No, no one's heard of mad cow. Okay. Mad cow disease is, yeah, we would catch it from eating contaminated beef. Okay. And it has to do with this protein. Right? There's a protein called the PRP protein, and it has two forms, the PRPC protein and then the PRPSC protein. Okay. The PRP protein is called prions. You can look it up if you want to read more about it. But prion proteins, you have it, I have it, cows have it. And if it's in this form where the protein structure is just helix, alpha helix, then it's fine. It's not going to cause any problem. We're not sure what it does, but it's not causing any problem. However, for cows with the mad cow disease, their proteins, the prion proteins, misfold themselves. And instead of having just the alpha helix, it now has these beta sheets in it as well. Okay? And here's the thing. The misfolded protein, the one with the beta sheets, it's like a bad influence, right? Once you have these misfolded proteins, it will go and find other normal proteins and will cause them to misfold. Right? So if let's say this green one here is the normal one, and then this is like a misfolded one, the red one. So the red one is gonna go and bind to the normal one, and then it's gonna force it to misfold, and before you know it, you're gonna have two of these red ones. Okay, and then they will go and cause other proteins to misfold. Right? And that's what happens. If you consume contaminated beef with the misfolded protein, it will cause your normal protein to start to misfold. And over time, if you have too much of these misfolded protein that builds up in your neurons in your brain, they cause the neuron to die, right? And it leaves behind these holes in your brain. This is the brain, okay? These holes in the brain, right? And that's what mad cow disease is. Okay, the full name is called spongiform encephalitis. Okay? Spongiform, you can tell from the name, it means your brain becomes like a sponge full of holes. And of course, it's deadly. Okay. It's not something you can like cook your beef completely and then it's going to be okay because it's a protein. Even if you cook it completely, you're not going to like kill it. Yeah, it's not living, right? It's not bacteria or viruses. So heat alone is not going to be able to kill it, right? And that's why they have to kill the cattle if they found out they have mad cow diseases. Okay, so that's one reason why understanding the alpha and the beta structure is important because in some cases it can cause disease. All right, so we're gonna move up to the next level. We're gonna move up to the next level, which is the tertiary structure. All right, I gotta make this thing again. So like I said, it's gonna form some loops in some places, and then it might have some zigzag here. I don't know if this thing is actually helping you. It might, it might not. So for the most part, this is 2D, right? 2D because you can put it on a piece of paper and it would lay there. Okay, nothing is like sticking out or anything like that. So that's a secondary structure. The tertiary structure is for this thing to actually fall on top of itself and then it forms a little ball here. It forms a little ball. Right? Now it's three-dimensional. Right? So the overall 3D structure of the protein is called the tertiary structure. Tertiary structure. And this tertiary structure is stabilized by the interaction between the side chains. Okay, so remember all those R groups, right? Different amino acids have different R groups over here, right? Some are non-polar, some are positive, some are negative. So the non-polar ones tend to find each other and bind to each other. The positive ones will be attracted to the negative ones, and so on and so forth. And they are able to form various interaction to help stabilize the overall three-dimensional structure. You do not have to memorize any of these, okay? The only thing you need to understand is how the tertiary structures are being stabilized. And finally, we have the quaternary structure. Not every proteins have this level of structures, only some do. So quaternary structure goes like this, right? Sometimes after forming like a 3D shape, this thing alone is not able to perform its full function. It needs like a partner, right? So like another one here, right? And together they will form a complex, right? Together they will be able to perform some functions, right? So that would be a quaternary structure. Which proteins they need to bring together to form a whole thing. So in this case, this particular protein has four smaller proteins in it. Okay. There's a polypeptide here and polypeptide here, two pink ones, two purple ones, and together they are able to form a complex and it allows it to perform its function. Okay. So let me summarize this for you a little bit. Hopefully after the summary, it will make you feel better. It's basically this chart. We can fill it in together. But I gotta copy it over first. Okay, this is question 17 in the study guide. If you haven't been working on the study guide, please do so. It's very important that you do. If you have trouble finding the study guide, you should let me know as well. I'll help you locate it. Okay, so 4 levels of protein structure, starting with the primary. Okay, primary is the sequence of amino acids determined by your DNA. And what holds them together would be the peptide bond. Okay, what's a peptide bond? It's right here. You should be able to identify a peptide bond. Okay, it's the CO and the NH, this one right here. It holds them together. You form a peptide bond by removing a water molecule. What does it look like? It's basically the order of amino acid. All right, so let me try to use some colors here for you. It's not letting me change color. Oh, too bad. We'll just keep going here. Okay, so that's like the first amino acid, second one, third one, fourth one, so on and so forth. They're all connected together. If I read the identity of each amino acid in order, that would be its primary sequence. Okay. Secondary. Okay, secondary is the peptide backbone forms alpha helix or beta sheets. Okay, beta pleated sheets or beta sheets. And the bonding characteristic, what holds them together? It would be the hydrogen bond of the peptide backbone. Right? So you might remember um, there are the alpha helices and then there are the zigzags, right? These are alpha, these are beta. You can have both. You don't have to have both. You might only have alpha, only have beta. All right. Tertiary. Okay. Tertiary is the overall 3D shape of the protein. What shape is it? Is it spherical? Is it globular? Is it tubular like a straw? And there are many different ways they could fold out. And the bonding characteristics, what holds the 3D shape together would be the interactions between the side chains, the side group. Okay. And again, it's going to form this three-dimensional shape with grooves in it and some bumps, right? And inside, it's, it's the all the structures that you have, the alpha and the beta and so on and so forth. I got to restart the program. 1 second. And finally, for quaternary, only remember only some proteins will go to this level. Right? This is when multiple polypeptide come together to form a complex. Right? So like you might have two red ones, and then you might have two blue ones coming together. Doesn't have to be two and two. It could be like whatever combination. Some of them has like 6, 7 different polypeptides coming together. But the point is they will work together. Okay, and how you hold them together would be interactions between the polypeptides. Okay, there would be some kind of interaction between the red one and the blue one that holds them together. All right. So like some interaction here, 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 here. Just staple them together, basically. Okay. Now these 3 levels, the secondary, tertiary, and the quaternary, these can be disrupted by heat, pH, or changes in salt concentration. Right? If you heat it up, the proteins is going to unfold. More on that later, but not the peptide bonds. Okay, peptide bonds are quite strong. Heating it, changing the pH is not going to break the peptide bond. Okay? Not easily broken. Any questions? All right. Hopefully you get it. I know it's a lot of stuff. Moving on. Importance of protein structure. Okay. Each level of protein structure contributes to the overall shape of the protein. The overall shape of the protein has another name. It's called conformation. Okay. Conformation. Conformation is shape, basically. Okay. So the shape of the protein is important for its function. It's a key and lock relationship. If you bend the key, if you change the shape of the key, it will no longer fit in the lock and you won't be able to open the lock, right? Similarly, if you alter the shape of the protein, then it will no longer work as it should. If the protein is responsible for performing some reactions in the body and you change the shape of the protein, then it will no longer be able to um, do that reaction for you. Okay. So what's the term that describes a protein structure being unraveled or disturbed? We say it is denatured or denaturation. Okay? So when a protein structure is altered. And there are a couple ways to do it. I already wrote it down for you earlier, right? You can heat things up or cool them down, changing the pH, right? That's why acidosis is dangerous. We talked about this last class, right? If your blood pH is too acidic, it will start to cause the proteins in your blood to unfold and denature, and they won't be able to do their function anymore, and that could be life-threatening. If you change the salt concentration or the type of environment that they're in, all these things would contribute to denaturing the protein. And it's not something you can reverse, okay? Think about cooking an egg. Where is the protein, by the way, in an egg? And sometimes people are not sure. Is it the egg white or the egg yolk?
The egg
Where
yolk.
the egg yolk, okay. Egg white, some people say white, some people say yolk. It's actually the white, okay? The white is where you'll find the protein. And yeah, the yolk is mostly fat, cholesterol, omega-3, that kind of stuff. So when you cook an egg, the egg white goes from transparent-ish to white, right? That's denaturation. You cannot uncook an egg, right? Once it's denatured, it's denatured. Same thing with cooking meat, right? Changing color, going from red to brown, that's denaturation. Okay? Meat is mostly protein, right? Okay? But remember, when you are denaturing the protein, you are only affecting the secondary, tertiary, and potentially quaternary structure. The primary structure is going to be okay. It's going to be unaffected. But doesn't matter. Once you misfold it, it's not going to function properly anymore. Cooling can also change it, okay? What kind of dessert has proteins in it and it's liquid, and after you cool it, it becomes kind of like a solid thing and it's not going to go back anymore. Do you guys know what I'm talking about? Sorry, I get now I say it out loud. A lot of things sound fits that description, right? I'm thinking about Jello. Okay, Jello is basically protein. and sugar, right? When you cool the Jello, you are denaturing the protein in the Jello, and it causes it to, solidify, right? If you make cake, you need to add gelatin, right? To help the cake set in some cases. If you're making like mousse cake and that kind of stuff, that's protein denaturing at low temperature. Okay? So generally speaking, you do not want to have your proteins denaturing in your body, right? Because you need the proteins to function the way they are. If you denature them, they're not gonna work properly, and it's not good for you. So let me give you an example where if you change the protein sequence, you end up having a disease. Okay? So there is something called sickle cell anemia. Okay, so anemia is a group of diseases, conditions where the person is not going to be able to deliver oxygen efficiently in their system. And there are different causes of anemia. You can have iron deficiency anemia. If you don't have enough iron, you will have anemia. This one is called sickle cell anemia. Okay. The protein that carries oxygen for you in the body is called hemoglobin. Okay, hemoglobin. Hemoglobin transport oxygen for you. And it's a protein that has 146 amino acid. If we look at the primary sequence, okay, it's valine, histidine, leucine, so on and so forth, right? Number 6 here, we have glutamic acid. You don't have to memorize the names, okay? I just happen to remember them. Now, what happens in someone who has sickle cell anemia? First of all, this is a genetic condition. You are born with it. It's not something you will catch, right? And there is a mistake in the DNA. We call that a mutation. So the instruction to make the protein is wrong. Instead of putting GLU in position number 6, the mistake caused your body to put VAL into position number 6 instead. Okay, so keep this in mind. We're looking at GLU and VAL. I'm going to go back. You don't have to. Okay, let's take a look at GLU. Where is GLU? Do I even have it on this thing right here? Okay, GLU is right here. Okay, GLU is a negatively charged amino acid. Okay, after the mutation, after the mistake, you replace GLU with VAL, which is a non-polar amino acid. So that's a pretty big change. The property of the amino acid changes, right? And that causes your hemoglobin to misfold, right? When the hemoglobin misfold, it actually causes your red blood cells. That's what these things are. Red blood cells. They're supposed to be circle in shape. But a person with sickle cell anemia, they would become banana shape, okay? Sickle shell, sickled shape, okay? Like a crescent moon here, right? And these things cannot transport oxygen. They actually get stuck in capillaries, and it's quite painful. And the person will have anemia. So even a single change in the amino acid is enough in some cases to cause severe disease. All right? Does this always happen? If you change one amino acid, is it always going to create a disease? Not necessarily. Okay, I'm going to go back here again. Right? Let's say if the mutation changes it from glutamic acid, GLU, to aspartic acid, ASP, then maybe it's okay. Maybe it's okay because look at it. Right? This is very similar to that. It just has an extra CH2, and they are both negatively charged. So if you are substituting the original amino acid with something that has similar property, then it's not as big of a deal. Okay, the function might be comparable. Okay, it might not even have an effect. All right. So we're going to talk more about these kind of mutations and their effects later on in lecture 7. All right. But for now, I just want you to understand the sequence of amino acid, which is the primary structure of the protein. It's important Because if it's wrong, then it will affect the secondary, tertiary, and ultimately the function of the protein. Does anybody have any question? Okay, we're almost there. Just a couple more slides. We will not be able to finish lecture two today. That's fine. We still have one more class this week before your test. What do we need proteins for? Proteins are very important in forming structures. Okay? Your muscles are made up of proteins. Nails, we don't have horns, but like the horns of animals are made up of proteins. Hair, spider webs, all these things are proteins. They also make up hormones. Some hormones are steroid, like the sex hormone. Others are protein hormones, like insulin. Okay. You might have heard of insulin. People with what kind of condition needs to have insulin in some cases? That's right, diabetes. Okay. Type 1 diabetes. People with type 1 diabetes, they need insulin, not so much type 2. Okay. But that's a hormone, that's a protein hormone. We also need protein for energy sources, right? We prefer sugar, but we also burn proteins as a way to get energy. And they also form something called enzymes. Okay, enzymes. We don't have time to go into these stories. Feel free to read them yourself, maybe another time, okay? I just want to spend the last couple of minutes telling you about enzymes, okay? So before I actually told you about enzymes, okay? People who are lactose intolerant, I told you they don't have the enzymes to break down lactose. What does it mean? First of all, enzymes are proteins, okay? Proteins. And they are proteins that are in your body, they're in the blood, they're in the cell sometimes, and they help catalyze chemical reactions. Catalyze basically means to speed up chemical reactions in the body. So a little bit of background information for you. Every single chemical reactions that take place needs to have a little bit of energy supplied to them first. Okay. You need to pay a little bit of energy before a reaction can happen. Okay. The amount of energy that you have to pay to start the reaction, that is called the EA, okay, EA, or activation activation energy. Okay. Again, it's the energy you have to pay before the reaction can happen. It's like paying your tuition before you can, attend the program. Now, that energy requirement is usually quite high, all right? And your body cannot pay the full price most of the time. And that's where an enzyme comes in, okay? The enzyme will help with the reaction by lowering the activation energy. Okay, you still have to pay, but the enzyme will make sure you don't pay as much. Okay, so it's like winning a scholarship. Right? Instead of paying, I don't know how much you guys pay, like instead of paying like $2,000 for a tuition, now you can attend by paying just 800. Okay, so the enzyme is gonna lower the activation energy and allow the reaction to happen faster and happens in the body without the high cost associated with it. Okay, so that's what an enzyme is. Basically, almost all the reaction that happens in the body will need to have an enzyme. Without the enzyme, you just don't have the energy to make them happen. For example, here is an enzyme. Okay, so this enzyme is a protein and it has what's called an active site. Okay, so active site, right here, active site is where the ingredients, okay, ingredients are also called substrates, where the ingredients of the reaction will fit. Okay. So here we have the active site and here are our substrate. I'm totally making this up. Let's say this is glucose and then this is uh, fructose. Okay. So these things will fit into the active site. The enzyme will help carry out the reaction by lowering the energy requirement, and then it will form the bond, hydrolysis in this case, and then you will get a product. Okay? So glucose plus fructose gives you sucrose. Okay? Without the help of the enzyme, sure, the reaction can still happen. It just won't happen in the body because it has a high energy requirement. Similarly, over here, let's say we have the sucrose. Okay, we have the sucrose. See, these are just kitchen sugar, right? And we know sucrose is made up of glucose and fructose. But the sugar that's sitting in your kitchen cabinet, they are not gonna spontaneously break down into glucose and fructose. Okay, because the energy requirement is not there, right? So when you eat them and put it in your body will have an enzyme that will take the sucrose, lower the energy requirement, lower the activation energy, and then it carries out the reaction and split out the product. That's basically what an enzyme does. Okay? Now, let's just say this is lactose. Okay? The enzyme that you need to break down lactose would be called lactase. And then it will break it into glucose and galactose. You don't have to write it down, okay? I'm just trying to make a point here. So what if the person does not have lactase? Remember, these are proteins and the proteins are determined by your DNA. And if your DNA is wrong or if it's changed, you might not be able to make the lactase. So without the lactase, you will eat the lactose from the ice cream, but you can't break it down. Instead, the bacteria will break it down and it's going to cause all those bloating and, gas that we talked about. All right. So enzymes are very important. Right? Anytime an enzyme is missing in the body, almost always you're going to have some kind of disease. Okay. I mean, lactose intolerant is fine. It's like, it's not life-threatening. But in other cases, if you're missing the enzyme to break down phenylalanine, right? Because you're of DNA mutation, then the person is going to be born with PKU. And if we don't know about it and feed them regular milk, then it's going to interfere with brain development, right? So that's why we need to, understand the different kinds of enzymes there are and the outcome if they are not working. Okay, sources of protein. Basically meat. Okay, all the meats have proteins. Dark meat, light meat, white meat doesn't matter. They all have proteins. Although red meat is a little bit not as healthy, they say. If you're vegetarian or vegan, then your go-to source for proteins would be beans, soy, nuts. They are all pretty good in proteins, like tofu, that kind of stuff, which is soy product, excellent sources of protein. And we only have two more questions, three, four questions here. Why don't you guys try it out? We can take it up later. But that is it for today. Please print out lecture three for next class as well. We'll finish up lecture two on Wednesday, and then we will start on number three. And also start studying for your quiz next Monday. Okay? If you cannot find things, just email me. All right? I'll tell you where to get them. You should be doing the lecture one and two practice quiz as well as lecture one and two study guides. Right? They will help you do better on the quiz. That's it. Thank you, guys. Take care. Stay warm.
Thank you. Have a good day.
You too.